I'm Fiona Brinkman. I'm a professor at Simon Fraser University in molecular biology and biochemistry, but I also have a cross appointment in computing science. I really enjoy using computers to study biology and in particular infectious diseases. And what we're doing is basically using computers to study the DNA code of infectious disease microbes and using that to understand how the diseases are spreading and better track them, sort of being like a DNA detective for infectious diseases. It's essentially a big data problem, so we're using Cedar to help us do this infectious disease tracking more quickly.
If you have a large volume of data, you go from a one-dimensional transaction-based data environment to a multi-dimensional large volume amount of data. You can start to look for patterns and do pattern recognition. And so it's growing knowledge based on understanding more and more about the depth and the breadth of the data.
You can bring people together around common research projects and link data in a way that wasn't possible before. We have researchers trying to make breakthroughs and needing computing resource, needing data management, needing to think about the processes for handling data in order to get that research done and to be competitive in the broader world outside of SFU.
The interconnect that we chose is the new Intel Omnipath architecture. And that allowed us to build a by far more powerful network architecture. We are running jobs on the system around the clock. So there are no idle resources really left on the system. So it shows the demand that researchers have for these kind of computing resources.
There's been a transformation in supercomputers and applications of more sophisticated methods for analysis that are allowing us to identify relationships between data that we hadn't seen before through things like machine learning approaches. And it's a very exciting time actually to be in this area because it's opening up a whole new world and there is so much data out there just waiting to be analyzed, and I really hope more people get excited about analyzing it.